Hi, this is, wait.
I'm first.
Hi, this is Miss Litton, and this is my wonderful 7th period AP Biology review. Say hi. Hi. Okay. So, a couple of things before. You're rude. Before we start our review today over Chapter 3, the chemistry of organic molecules, which I'm super excited about, I want to address something with you on your calendar for a momento. Okay? So we have tomorrow, right? And tomorrow will be 8:21. Will we be finishing Chapter 4?
We'll be starting it.
You'll be starting it in your class period. What period is that?
First, second, third.
No, first and third already started it, and actually fifth did too. But you should engage more. But we will be finishing up Chapter 4. Here's the only thing is, this is your first test, and I'm afraid you're a little nervous about it, potentially, and it's on a Monday morning. I can't give, you can see every day is scheduled for the whole semester. I have no days to give you. But I am willing to do this if you're interested. I can do tradesies. And what I can trade out is I can trade out Monday's exam for starting Chapter 5, membrane structure. And if we trade that out, Monday I will start Chapter 5, membrane structure, and that will give you another couple nights to maybe ask me questions so you're ready to go on Wednesday. How are you thinking about that? And I'm going to pause just for a minute for discussion. Pause. All right, so we just took a few minutes to discuss, so now we're going to take a vote. So all those who want to switch Monday and Wednesday on the calendar, say aye. Aye. And those who do not, say nay. Nay. I'm going to say probably the ayes outweigh the nays. Nays, what can I do to help you if you have a nay? What was the issue that you're worried about?
I don't want to learn something new before they do.
You don't want to learn something new?
Yeah.
Okay, so here's the deal. You have to know it all by the semester final, and there's always ghost questions. So if your learning is like, I can't take anything more until I have it, that's not a really good way to approach it. So I'm going to tell you it's probably good that I'm doing this. Okay, because that's not the approach I'm going to take. Yes?
I want to get it over with.
You want to get it over with. That I can understand. That's a good argument. You want to get it over with. And I'm willing, it's no problem, we can have the test on Monday, but I took the vote. I think there's more that would appreciate the extra day, so I'm going to go ahead and give that extra day. Just going with the vote. Okay? All right. Now, all right. Come back to me. So a couple things about the chemistry of organic molecules. You do not need to memorize all of the functional groups. I just like you to be aware of them so you can understand, like when we're doing dehydration synthesis, you understand what has to happen, that you're going to take an OH and an H, those types of things. Some of these functional groups are going to come up again and again. Something like a carboxyl group, I want you to get in the habit of realizing when you have that as a functional group, oftentimes you're going to lose that what? Hydrogen, yeah. Okay, things like that. I want you to recognize that some of those groups are polar, and if they're polar, then they're hydrophilic. Yeah, and so they will work well in water. And we talked about the difference between hydrophilic and hydrophobic. And then the next thing is we talked about the idea of monomers and polymers. And in your mind, if you want to know, do I understand this chapter, or do I know the content of it, then you would be able to make yourself, go home and make yourself a chart, just like that chart I showed you, where you have monomers, right? And you have polymers and structure and function. So if you can just kind of whip that out and draw a little sketch of each one, that would be a good place to be as far as knowing this content, okay? So we know our 4 important organic molecules are carbohydrates. And if I was going to say, what are carbohydrates built out of, what would you tell me? You would tell me sugars, okay? So if you said sugars, what would you say would be a good way to recognize a sugar in a diagram? Could you draw it, like in your mind? Something like that?
Yeah.
You see it? And what is that representing? A simple sugar. Yeah. All right? So that's what carbohydrates are built out of, and then that's like the building block for them. Lipids we had two categories, right? What were our two categories of lipids? Yeah, we had steroids. Yeah, and we had our fats, or what's called triglycerides. Yeah. If I was going to do a simple drawing of a fat so I remembered it, what would you do? You're thinking something in your head, so if you recognize it, if it came up on a test. Show me with your fingers how you would draw it. I like the way you draw. Right? Or you could go like this, something like this. Yeah? Okay? So what does the this represent? What's the that? That's glycerol. Then what would I have here? That's right. Okay? Now, the way in which I've drawn it, you would assume that those fatty acid chains were what? Saturated. So you could put something on there if you wanted to. You could say sat, unsat, to remind yourself about that. You could label this as glycerol. Okay, and you would want to be able to identify what are some of the functions, what do they do? And we looked at an example. We said long-term energy storage, but then we said we could build a what off of this? What could we build? It was an example. It's really important. Phospholipid. And in a phospholipid, in a phospholipid, what do? You remove one of the fatty acid chains and you replace it with a phosphate group. Now all of a sudden, our important organic molecule that was hydrophobic, we now have a region of it that's hydrophilic. Okay? And so that is what, it's 2 layers of phospholipids that builds our cell membrane. Okay? Now, if I wanted to do steroids, what would I do for steroids?
4 fused.
Yeah, 4 fused. And I mean, you could just do something simple so you remember them. Okay? And we would talk about hormones, and hormones are derived from cholesterol, and we would talk about how they're chemical messengers. The cholesterol is in the structure of cell membranes. We would just have a few things about it. Okay? For proteins, what's the building block of proteins?
Amino acids.
Amino acids. Okay, I want you to imagine an amino acid structure right now in your brain. Don't say it out loud. Don't whisper it to your bio buddy. Just think it in your head. If you need to draw it so you can imitate it. But don't show it to somebody. Nobody's going to be impressed if you can show them before they can think it. They'll just be annoyed. Okay, what would you start out with if you're going to draw an amino acid? Exactly. Then what would I put? Because I know the way carbon acts. I do 4 bonds. What could I put up top? What I put down below? How many different ones of those are there? 20. Some are hydrophilic, some are hydrophobic, right? What would I put over here? Yep. And it might become a NH3. What would I put over here? C double bond O, OH. Carboxyl group, yeah. Carboxyl group, amino group, hydrogen, and R group. That's exactly what I would do. And I would know that when I hook one amino acid to another amino acid, just like we learned before, what's that reaction called? Dehydration. Synthesis. What's another name for that? Condensation. Synthesis. What's the name of the bond specifically between amino acids? Peptide bond. Peptide bond. And that's why oftentimes they're referred to as polypeptides. Okay? Then I would know what are some functions of proteins. We said yes, you can find them in muscles, but where else could we find it? What are some other functions of proteins we thought were important? Yes?
They can be a carrier.
They could be a carrier in a cell membrane. Yes, muscle, I said that. Give me another one. Metabolism, how? They are what? Starts with an E. Enzymes. Right? Enzymes, cell communication. What else? Yes. Yes, you can find it in hemoglobin. Yes, you want to think, and that would be in your blood. Okay, so you want to think about what are all the functions of enzymes. Yes.
Also, would we, for the DNA, they make their the helper protein? What was it called?
chaperone proteins.
Yeah.
Help fold enzymes. I like that. Helper proteins, we got it. Chaperone. Good, so you just want to be familiar with that. We have a little more on that. Nucleic acids, what are they built out of? Nucleotides. Okay? And so nucleotides consist of what three things? Okay, a 5-carbon sugar. What else?
A base.
Yep, a base. As I've drawn it there, it would be what kind of base?
Nitrogen.
Yeah, it is a nitrogenous base. You're right. 2 rings. It's a purine, hydropurine. And what else would you have? A phosphate group. Okay, that's a nucleotide. And if you recall from our diagram, we know how many different bases are there? 5. Okay, 3 of them are pyrimidines. 2 of them are purines. Okay, so we have 5 bases to choose from. What base you have, is it going to be dependent on whether you're DNA or RNA?
Yes.
What bases could I find in DNA? Adenine, thymine, and in fact, in DNA, adenine, because DNA is two chains, right? It's 2 chains, so you would have adenine in a hydrogen bond with thymine. How many hydrogen bonds? 2. Okay, what else could I find? Guanine and cytosine. And how many hydrogen bonds? 3. Okay, now connected to one of those 4 bases, besides the hydrogen bond that it has with the nitrogenous base of the second chain, what else would it be connected to? What else would that nitrogenous base be connected to? What? Oh, you're saying it. I think you're saying it. Deoxyribose, which is your 5-carbon sugar you would find in DNA. Right? I would have deoxyribose. I wouldn't find a ribose connected to thymine, right? Right, I would have to have a uracil. You understand that? Okay, because you only find deoxyribose in DNA, hence the name DNA, deoxyribose. And then RNA is the only place you find ribose. Good. Now that sugar, it's connected to a nitrogenous base, yes? What's above it and below it? Phosphate group. It's above and below it. Yeah, phosphate group. So start to get the structure in your head when you imagine it. If you think of DNA like a ladder, the sides of the ladder, like there's the rungs of the ladder, I would crawl up. Those would be the nitrogenous bases with hydrogen bonds with each other, right? That would be the rungs. The sides of the ladder would be alternating what? Phosphates and sugars. Okay, and we can look at a picture of that to refresh your memory. Yes?
So just making sure the 2 sugars that connect to the base would be the deoxy and then the ribose.
Yeah, but ribose in RNA and deoxyribose in DNA. And the phosphate's the same. Okay, and you always have a purine will be connected to a pyrimidine, and it keeps the diameter the same on DNA. So I think we need to look at a couple pictures on that because you were slow to that explanation. Okay, so we have here 5 different bases. How many different sugars? 2. And we have 1 kind of phosphate. All right, so looking through, I'm just going to jump to DNA because that seemed the part that seemed so hardest. Okay, come back to me. I need to be the MIP in the room. Okay, so here we can see the 5-carbon sugar. Here we can see a nitrogenous base. Is this nitrogenous base a purine or a pyrimidine? Pyrimidine. I know it's a pyrimidine because it only has 1 ring. If this were in DNA, it would be bound to a purine, because a pyrimidine and a purine, and I would know that because in DNA there's always 2 strands, whereas in RNA there's 1. Okay? So I have a base, a sugar, and a phosphate. Okay? Here are your 2 sugars. We talked about that, deoxyribose and ribose. Here are 5 nitrogenous bases. But when we look at DNA right here, okay, this is like a ladder. Can you imagine that as a ladder? Okay, and this is just a twisted ladder. which would be a great band name in the future. Okay, so if you look right here, you can see the 2 hydrogen bonds between adenine and thymine, 2 hydrogen bonds, thymine, adenine, 3 between guanine and cytosine. If I look at the edges, the part that's outlined in blue, those are alternating what? Sugars and phosphates. Okay? Now, the only thing a phosphate is bound to is what? A sugar, right, above it, and a sugar below it. But the sugar is bound to 2 phosphates and a nitrogenous base. And a nitrogenous base is bound to a sugar and then hydrogen bonds between it and another nitrogenous base, which is connected to a sugar, which has above and below it phosphates. Got it? Okay? Flow it together. Flow it together. Okay? Whereas RNA is just a single strand. It's like half the ladder. And it's just alternating what? Phosphates and sugars, but coming off the sugars are nitrogenous bases. Same nitrogenous bases as found as what's in DNA, except, yeah, there's no thymine, instead there is uracil. And then here's another picture. You can see the nitrogenous bases and the hydrogen bonds between them, and then the sugars alternating phosphates. Okay? And then here's that summary diagram, which you all have, and I also put it on my Instagram. Yes?
There's something I had a question about. I remember I was looking at it yesterday. It was like there's something that was in a monomer and in a polymer. Steroids, yeah.
Yeah, steroids is weird. It doesn't really fall. The lipids are kind of the unusual one, because it doesn't really just fall like monomer, polymer, because steroids are 4 rings of carbon. So you don't really break it down into monomer, polymer.
So are fatty acids and glycerol, are they not monomers?
No, we would call that's the monomer to build glycerides, triglycerides, yeah.
Only triglycerides or just any type of lipid?
Well, lipids have 2 categories, fats and steroids, so not any kind of lipid. Yeah, the lipid umbrella, hiding underneath the lipid umbrella are fats and steroids. Steroids are always going to be 4 rings of carbon. Fats are going to have glycerol and fatty acid chains. And we also have waxes, which are long-chain fatty acids connected to long-chain alcohols. And we also have the modification of phospholipids, where you remove one of the fatty acid chains and put a phosphate.
Yes.
Everybody knows this, okay? Other questions? Do you need me to go over lipids again?
Yeah.
Okay, let's do it. I also have a review game. I have a different review game for you. To everything over, restart. But look, you guys, did I remember to push yes, we have 18 minutes of fun right here, and I will load that on my YouTube page so you can watch it, while you're eating dinner, eating tacos tonight, maybe? I don't know. I don't know, that sounds good to me right now. See, look at me. I love you, but we need to focus. We don't have time to meditate right now, self-meditate. Okay. So, we have fats and we have oils. They are built the same in the sense that they both have what? Glycerol and fatty acid chains. It's just that when you have oils, their fatty acid chains are going to be what? Unsaturated. And fats, when we think of animal fats, they're going to be what? Saturated. But really, oil is a fat, right? It's just a liquid one. Okay. Phospholipids, either I'm going to talk or you're going to talk, but we both can't do it because it's annoying. Okay? So phospholipids are exactly the same as that. You still have glycerol, you still have fatty acid chains, but instead of that 3rd fatty acid chain, what do we have? Yeah, we have a phosphate group. And this is where you would find those in cell membranes. Steroids are going to be 4 rings, right? That's a steroid. And then when we look at waxes have a fatty acid, which just has that same ending group, functional group that you have here. But then it's bound to, I need some other color, it's bound to an alcohol. Same fatty acid chain, but it has an alcohol on the end. So that's why it's kind of, what you have in there a lot is carbon and hydrogen, carbon and hydrogen, carbon and hydrogen, carbon and hydrogen, carbon and hydrogen. Okay, a lot of that. Yes.
I have a little obvious, but in phospholipids, I'm sorry, I need to like pull myself together. In phospholipids, are the fatty acid chains unsaturated or saturated?
It depends on if it's a saturated fat or an unsaturated fat.
Okay.
Yeah. So remember, saturated means that that chain, the long fatty acid chain, has all single bonds between the carbons, and then the hydrogens are like, it's saturated with hydrogens. But in unsaturated fat, you have some, double bonds in there, which then puts kinks in the chains, which helps it to be a liquid at room temperature. All right? You good on that? Feel better? Yes.
You said there's an exception to the.
Oh, palm oil. I just feel the need to say that in class, but I don't think anyone's ever going to ask you that, but I feel like, like I'll say, you are made out of eukaryotic cells, you have membrane enclosed organelles, and you all have a nucleus except mature red blood cells because they make more room for hemoglobin. So I'm just giving you the exception. There's always exceptions. Yes?
Sorry, so for laxatives, a fatty acid chain connected to it.
It's like two fatty acids, long fatty acid chains. That part's the same. It's just the end functional group is different.
Okay.
That's all. Okay, so those are lipids. Do we need to spend any more time on carbohydrates? You pretty good with carbohydrates? Do you remember this part right here? Polysaccharides fall under 2, right? Who were our energy storage ones? Starch and glycogen. Okay. Who are our structural ones?
There's a bacteria.
And what else?
Not bacteria.
That's all you need to know. Okay, we looked, here are monosaccharides joined together to form disaccharides, right? So sucrose is glucose plus fructose, maltose is glucose plus glucose, and lactose is glucose plus lactose. And those are your monosaccharides. That part's easy. Now, all right. The third nucleic acid. The third nucleic acid that I didn't mention earlier that I need to mention. Where are those? Where's that chart? Here it is. Nucleic acids. When we talk about our examples, we have DNA. What else? RNA and ATP. That's energy. Okay, ATP is for energy. So I just want to mention that. Okay, I think I have a monster game, and I also have some. Look, this is before I did it digitally. Do you understand it?
Yes.
I did that in a review.
I mean, okay, I understand it now, but like if you
Yeah,
taught
but
it like
you should
this,
understand it.
if you taught
I
it
did
like
it in a
this,
review, yeah, if I taught it like, we were just reviewing and but guys, let me tell you, this is something you could do, right? You can see I hand wrote it, I was sloppy just summarizing, but we were in 7th period review and I go, let's write down what we know. So we did this collaboratively. We made this little box, we drew it out. You can recognize, look in there, sugars, look, structure, energy, you know this, glycerol, fatty acids, saturated, unsaturated, phospholipids, steroids, primary, secondary, tertiary, quaternary structure, you understand that. Peptide bonds, nucleotides, here are where you find them, how many hydrogen bases. That should be something that you can just pull out of your brain, okay? I think I have a monster quiz here. You can get into teams. Oh wait, let's wait. That's the class ID. So you can go in 85277, and then you can go in as whatever you want to go in as. And I'm going to divide you into teams. Hurry up, you've got like 1 minute. Less than 1 minute. I'm going to pause you for a moment. Okay. I turned the recording back on just so you can see the answers in the review. Okay.
1 second and our thing's better.
Okay, so everybody got the first one. That's great. Hydrolysis reaction brings two monomers together. Is that true? What does hydrolysis do? It breaks apart. Which of the following are energy storage molecules? Starch and glycogen. Phospholipids make up cell membranes. True. Amino acids are the monomers for nucleic acids. No. What is the monomer for nucleic acids? Nucleotides. Saturated fats do not have double bonds between the carbons. True. The secondary structure of proteins depends upon the R groups of the amino acids, charged, polar, and yeah, but look at how many of you said true. Okay, so hear me. The secondary structure is just due to the what? Charge amino and carboxyl group charges, right? The tertiary structure is due to the R group. Okay. Uracil is found in RNA only. Adenine is found in, whoa. What's the only ones that are unique? Thymine in DNA and uracil in RNA. Okay. And is ATP an energy storage molecule? Yes, true. Okay, so that's it on that. I think I also have this. If anybody else wants to log in, I'm going to pause this for a minute and then we'll just go over the answers. They're still working on this practice quiz. I'll show you the results in a minute, but I want to remind you, do your highly suggested reading and thinking, and don't forget you could have a ghost question or two. So here we go. We're going to go over your answers. Nucleic acids are built out of nucleotides. Amino acids are build proteins. Guys, can we do this together and then you can talk afterwards? 4 rings of carbon will build steroids. Sugars like glucose build carbohydrates. Nucleotides build nucleic acids, not proteins. Oh! When you do dehydration synthesis using, what does MAP stand for? Multiple answers possible. Okay, so stop, guys. So when you're doing dehydration synthesis, the first thing I would do if I saw a question like that is I would run through my head, what is dehydration synthesis? Is it about building or breaking? I'm going to be building things. So now I need to assume that the statements below need to have something to do about building, and are they building the right thing? Because you're going to take monomers and build what? Polymers. So amino acids to form a protein? Yes. That's building using monomers to polymers. Amino acids to form a water molecule, will that happen? Yes. Through the what process? Dehydration synthesis. You'll form water. Fatty acids and glycerol to form a nucleic acid? No. Okay, because fatty acids and glycerol will form what? Yeah, triglycerides or fats or oils. Am I going to do polymers and form monomers? No, what would the name of that reaction be called? Hydrolysis. Nucleotides to form a nucleic acid? Yes. And then the last one is when you do hydrolysis to a fat, you produce. I have a fat. What's a fat made out of? Glycerol and fatty acids. So I'm going to produce what? Glycerol and fatty acids. That one is terrifying. And which of the following? I taught 3 classes today. I have not had a break. I had a meeting at lunch. So when I'm helping you, don't make me talk over the top of you, okay? Because I have not stopped today. So when you do the following to a DNA nucleotide, what are you going to have? Deoxyribose, adenine, phosphate. Why? 2 of you picked A. What's wrong with A? There's a uracil. What's wrong with B? Ribose. What's wrong with C? Yeah, 2 things against you. And what's wrong with D? Ribose. Okay? All right? Best strategies. Remember, you can read through your group shared notes. You can read the summary I gave you at the end. That's all input studying. Don't forget to do output studying. You have a practice quiz on there too, right? Highly suggested reading and thinking. Make good choices. You're smart and I love you.